Your Excellency, Mr. Governor, Nobel Laureates, students and colleagues, ladies and gentlemen, welcome to Uppsala University and to our grand auditorium. Dear Nobel Laureates, thank you for coming here today. Thank you for your contributions to science. And thank you for helping us uphold this great and important tradition. For more than 140 years, this auditorium has been the place where the university celebrates and honors international leaders and role models. This is a forum for meeting the Uppsala University audience. To set the standard, Demosthenes, the orator, and Sophocles, the poet, are placed at either side of the stage. They stand here to illustrate the fact that we take our place in a long tradition of knowledge and that we have a heritage to manage. Even now, they have plenty to tell us. Two quotations are on my mind today. Demosthenes' drastic saying, "All speech is vain and empty unless it be accompanied by action." And Sophocles' conclusion, "Our happiness depends on wisdom all the way." If we put them together, we could say that our happiness depends on wisdom manifested in action. To my mind, you, Nobel laureates, are ambassadors for this way of thinking. Your knowledge and your wisdom have crystallized into practical form and yielded results that have enhanced our knowledge. And this has now been recognized and rewarded. Our primary goal at Uppsala University is to contribute to a better world. By your determined efforts, you have shown that this is possible. Knowledge and enlightenment enable us to understand our world. As we together find ways to turn knowledge into action, step by step, we solve challenges confronting humankind and the world. In this way, we will attain the happiness Sophocles speaks of. Again, I would like to thank you for coming here today. Thank you.
Uppsala University is this year extremely happy and honored to be able to present 4 of the Nobel Prize winners this year. And it's my real pleasure to introduce them to you. My name is Jonas Bergqvist. I'm the professor of the Department of Chemistry at the Biomedical Center here at Uppsala, but also the chairman of the Chemical Society in Uppsala. And before I introduce our dear Nobel laureates, I will ask them all to make favor to science and education and help me to share a prize to the best chemistry student this year in Uppsala. This year, the prize has been awarded to Sofia Lindblad. And please, Sofia, can you join us at the stage, please? Sofia has presented an excellent master thesis together with her supervisor, Professor Mate Erdely. Congratulations, Mate, also for your contribution. And they published this paper in JACS. which is a very prestigious journal. So please, Nobel Laureates, can I ask you for some assistance? We have a diploma signed by all 4 Nobel Laureates, and we have a bunch of money that's going to be handed over.
Better not give it to us.
Okay, I'll pass it on to Donna then. So.
Gratis.
Shake the hand. Grattis. Congratulations. Thank you so much. Thank you. And congratulations again. So, first out of the Nobel lecturers will be Professor George Smith of the University of Missouri. So Professor Smith has received the Nobel Prize in Chemistry for developing an ingenious and elegant method known as the phage display, and we soon learn what that is. That's when you take a virus that infects a bacteria with a bacteriophage, and then that can be used to produce new proteins. Please join me in welcoming Professor Smith at the stage.
Thank you very much. I'm honored to be here in this wonderful auditorium. And I'm going to try in a very short time to tell you about phage display and how surface plasmon resonance, the vehicle, was developed at this university, giving rise to a company called Biocor. So, phage display. A phage is a virus that infects bacteria. I'm probably not enlightening anyone by that statement. Here's an electron micrograph of the particular kind of phage that phage display was developed with. Filamentous phage because of their long, thin, filament-like shape. On the left is a cartoon used to represent it. On the outside of the phage particle is a coat made of phage coat proteins, and then on the inside is the phage chromosome with the phage genes. I was particularly interested in a particular coat protein gene that codes for a protein that's displayed on the surface of the phage particle at the tip. A phage display construct is one in which a foreign piece of DNA, represented by the purple rectangle, is inserted into that coat protein gene. And so the amino acids encoded by that foreign piece of DNA are displayed on the surface just like the coat protein itself is. So I'm representing that here by these purple round-shaped peptides. So for the rest of this talk, I'm going to abbreviate that cartoon by leaving out everything inside the phage except the little piece of colored DNA. So I'm going to first describe a random peptide library. So a random peptide library is a huge collection of phage clones. Say 250 million, and a modern one might be 100 billion. Each clone has a different short piece of DNA in it, enough, in this case, to encode 15 amino acid. And so each clone displays a different 15 amino acid peptide. And they're random peptides. They don't have any particular origin. They're just random. So there's blue round peptides and sharp gold peptides and another clone with square green peptides. Here's a picture of a library. It has about 10 to the 15th phage particles. This is an early one representing about 250 million clones, so 250 million different 15 amino acid peptides. So I'm now going to describe affinity selection. So I'm going to use to illustrate it a particular model system that we use in our laboratory several times to develop phage display technology. So we start with a receptor. It was a commercially available receptor, but we considered a model for perhaps a receptor that's involved in disease. And a graduate student of mine, Jinan Yu, had a task of finding in that bottle with 250 million peptides in it, very rare peptides that would happen to have the ability to bind strongly to that receptor. Obviously, this is a really hard task if she had to go one at a time through all those, but she didn't have to, and that's where affinity selection comes in. So the first step in affinity selection is to immobilize the receptor on a solid surface. In her case, it was just the plastic surface of a petri dish. Secondly, she poured the library on it. Each clone in the library is represented by tens of thousands of different particles, not different particles, identical particles. So the library is poured onto the dish. During that period of exposure to the surface of the dish, any phage particle whose displayed peptide has affinity for the immobilized receptor is captured on the surface. All the unbound phages can be washed away, and then the phage that you want, the one whose peptide happens to bind to that receptor, can be released from the surface of from the solid surface. But we're not home free yet, because the phage particles that are released in this way are in tiny numbers for two reasons. One, any one phage clone is just a tiny fraction of all the phages that were originally put in the dish. And secondly, this capture process is very inefficient. At maximum, something like 1% efficiency of capture. So the number of particles released is tiny, way too few to analyze, but it doesn't matter because these phages are infective. Simply by infecting bacteria, we can grow unlimited numbers of them, trillions or 1,000 trillion if we need to. This is the essence of affinity selection, allowing you to search through huge libraries of structures for very rare ones that have the desired activity. So let me use an abstract way of representing this process. We construct a very large library with a huge number of phage particles, representing a huge number of different structures. So that's the initial population. We subject that population to affinity selection, discard the ones that don't meet the criterion for selection, and leaving a fittest subpopulation, but it's a tiny fittest subpopulation, but it doesn't matter because that population can be expanded enormously simply by virtue of infecting fresh bacterial cells. And as also represented on this slide, the amplified subpopulation with now trillions of particles, generally after one round, is not purely just the peptides that are of interest. They're mixed with other ones because the discrimination isn't perfect. So typically we have to go through additional rounds of affinity selection. And finally, after the final round, the peptides that are on those phage particles are analyzed. So I'm just giving a list of the peptides that emerge from this kind of selection. And I'm going to emphasize here that the different peptides I've shown here have different affinities for the receptor as determined independently. So there's a top one here, I'm calling it a champion, and that's just the word we use, and it doesn't mean anything in this context. And it has a KD of 500 nanomolar. So KD, just to give the shorthand, KD, if it's high, it's weak binding. If it's low, it's very strong binding. So this is not a very good binder, 500 nanomolar KD. But there are two other peptides that emerged from this experiment. We're calling it J72 and J88 with considerably higher affinities. And from a previous experiment, a 6 amino acid peptide with a KD of 3,000 nanomolar, so that's terrible affinity. And then finally a control phage that has no affinity at all. So what I'm going to describe now is experiments using surface plasmon resonance, developed here at Uppsala University and by the Biacor company, also in Uppsala. So here, the solid surface that we're representing is a center chip. And the center chip has these chains on them to which a receptor can be attached covalently. That receptor in our case was that receptor that we've just been talking about, but in SPR, or surface plasmon resonance terminology, that would be called the ligand. And then another molecule called the analyte, which isn't bound to anything, is flowed over the surface of that chip. And when the ligand binds to, when the analyte binds to the ligand, it increases the refractive index at the surface. And in surface plasmon resonance, that can be very sensitively detected in real time. So that is the essence of surface plasmon resonance. So in our case, this is an analog of affinity selection. The solid surface isn't a petri dish, it's a sensor chip. And the first step in this experiment was to covalently attach to that solid surface the molecule streptavidin, which has extremely high affinity for the biotin, for the vitamin biotin. In the second step, then, the ligand in SPR terms, and that's the receptor, our model receptor, is flowed over this chip, and the receptor has biotin attached, so it's immobilized by virtue of the biotin binding to the streptavidin. And the third step, we flow phages as the analyte over this chip. And the phages as the phages bind to the receptor, the refractive index increases, and that signal is recorded. So here's a diagram of how 4 flow cells are present on a single chip. We have 4 independently addressable flow cells. So in the first step that I described, where streptavidin is attached, streptavidin is flowed through all 4 flow cells in large amounts, so large amounts are immobilized on the surface. And the refractive index increase is gauged by these resonance units, or RU. So that 1,000 is a lot of resonance units. We've attached a lot of streptavidin. That's at the top. Then in the next step, the biotinylated receptor is flowed through the flow cells one at a time. In one, no biotinylated receptor. In the second one, a little bit, so only 3 RUs are immobilized. In the third one, a little bit more, so 18 RUs are immobilized. And the last one, 300 RUs are immobilized. So this is an analog, we hope, of increasing stringency of selection. Binding to the 3 RU, the flow cell 2, means you need really strong affinity because the immobilized receptor is rare. And that contrasts with the last one, where there's 300 RUs worth of receptor immobilized, so that so even low affinity ones may be able to bind to that. So finally, in the last stage, the phages, 5 different kinds of phages with different affinities for the receptor are flowed through. They are flowed through these in series. First through the control flow cell where there's no receptor, and in turn through the other 4. And during that time, the signal is recorded, and that gives rise to a centrogram. Let's show the results for the control phage. So on the top signal, the one that you're seeing on the top there, the control phage in the first channel, its signal is shown. And this is the signal that doesn't indicate any binding. There's a sharp increase. It's because the refractive index of the solution of phage is slightly different from that of the previous buffer that was flowing through the flow cell. So there's a slight increase there. And then there's a gradual, very small increase during that time. And then when we switch back to the wash buffer, the refractive index decreases. So in the other 3 flow cells, that signal from the first one is subtracted from the others. And you can see on the bottom curve, all 3 of those centrogram signals are essentially 0. So that means that we are seeing no signal when there shouldn't be any binding. So let me show the centrogram for the weakest binder, the one with 3,000 nanomolar. You can see that we have a pretty strong signal with the greatest amount of receptor, an intermediate signal with the intermediate amount of receptor, and very small signal with the very low density receptor. The same pattern we're seeing on all of them. The champion peptide, 500 nanomolar, here's 30 nanomolar, here's 12 nanomolar. Now, here I'm showing them all 5 different phages together on the same centrogram. And you can see the control phage, that's the red signal on the bottom, gives no signal, but the other 4 that do bind, all 4 of them with the highest density of receptor are giving essentially the same signal. At the intermediate density of receptor, you can see that the 2 best binders are indistinguishable and high. The next one down is much weaker, and again, the control is essentially 0. And here's the lowest density receptor, and here you can see there's really strong discrimination between the best binder in the light blue curve and the others. So this is showing, the conclusion of this is that the stringency of selection can be adjusted simply by controlling the density of the ligand, in the target receptor in our experiment on the surface. So the question is, does this mean we should use that very low ligand concentration right from the start? And the answer to that question is no, we shouldn't. Because let's look at the very best binder at the 3 different levels of stringency. You can see at the least stringency, the high curve, there's a lot more capture than in intermediate stringency and at high stringency. And that's very important that in the first round of selection that you capture as many as you can, because remember when I was talking about affinity selection, the affinity selection at best is like 1%. But if we're making that like 0.1% or 0.01% by using high stringency, we'll miss even the best binder, and we can never recover it. So the strategy that comes from this is that at the very first round of selection, the stringency should be very high, and at the following ones, we can increase stringency to try to capture the very best binders. So thank you very much.
Thank you so much. Our next speaker is Professor Sir Gregory Winter at the MRC Laboratory of Molecular Biology in Cambridge, England. Sir Gregory Winter has received the Nobel Prize in Chemistry for his work on the Phage display of autoantibodies. His use of direct evolution of antibodies has led to new pharmaceuticals treating autoimmune diseases and metastatic cancers, among other things. So please join me in welcoming Sir Gregory Winter.
Well, good morning, ladies and gentlemen. It's a pleasure to be here, and I would like to thank the university and the Vice-Chancellor for inviting me. I'm going to talk about harnessing evolution to make medicines. My affiliations are the Medical Research Council, Laboratory of Molecular Biology, and also the Center for Protein Engineering, and much of the work was done with the company Cambridge Antibody Technology, which I founded with a couple of other scientists in the early 90s. So let me tell you about antibodies. Antibodies are proteins that form part of our normal immune defense system. They're responsible for binding to viruses and bacteria. And they act in two ways. First of all, the end of the molecule, if you imagine it's a Y-shaped molecule, the end of the arms of the Y bind to the target virus or bacterium. At these tips of the Y, you see a series of, there's a scaffold on which is built a series of loops. And between one antibody and another, I'm going to have to use this thing. So where's it gone? Oh, here. Okay. So it can't, no, it's hopeless. Right. So on the tips of the Y, you have this scaffold of what's called beta sheet, and we have a series of 6 loops mounted on that. In each different antibody, those loops can be of different sequence. In fact, they invariably are of different sequence, and that means that antibodies can bind a wide range of different targets. The binding to a target is, in many cases, quite sufficient to block its activity. It can block a virus from engaging with cell receptors. But the antibodies have another trick, which is the other end of the molecule acts as a flag for the immune system, and it recruits several different kinds of immune cells to kill whatever the antibody is attached to. So these two different mechanisms, binding, blocking, and killing directly, are important activities of the antibody. Now, although antibodies were primarily designed and have evolved to be against infectious disease, the major use in modern medicine of them has been in treating non-infectious disease, such as cancer and autoimmune disease. If we look now at the best-selling medicines in 2016, I've marked in red those medicines which are antibodies, and in black those which are standard chemical pharmaceuticals. And what you'll see is of the top 8 products, that 6 are antibodies and 2 are chemicals. Now, you probably thought of pharmaceuticals as being chemicals. Certainly in the top-selling ranks, they are now antibodies. And all of those antibodies are used for treating non-infectious disease, such as cancer and autoimmune disease. Now, that didn't happen overnight. It took quite a few years to develop antibodies capable of doing this. And the big issue is that the immune system is attuned to recognize foreign molecules, and therefore scientists had to work out a way of trying to get human beings to make antibodies against human proteins such as would distinguish a normal cell from a cancer cell. And there are two ways of doing this. The first approach involved taking, for example, taking a mouse, immunizing a mouse with a human cancer, and this generates mouse antibodies. And there's a technology called hybridoma technology, which allows you to make individual mouse antibodies against those cancer cells. But if you take those mouse antibodies and you put them into humans, they do actually kill the tumor. The problem is, after about 10 days, they stop working. And the reason is, the humans see the mouse antibodies as foreign and attack the mouse antibodies and block their activities. So ideally, we want to make human antibodies against human targets. And as we know, humans don't make it, and obviously mice make mice antibodies. So therefore, we're using genetic engineering. A variety of groups, ourselves included, started to gradually take those mouse antibodies and to humanize them. And we can see here, the first one is called a chimeric antibody, where we take the regions, the antigen binding regions at the tips of the Y, and we transplant those onto a white human antibody. And the stage beyond that is we take those loops, which just bind to the antigen, those 6 loops, and we can just transplant those into a human antibody. And that's the ultimate in humanizing an antibody. And this was a technology we developed in the mid-80s, and this has given rise to a number of very effective pharmaceuticals, including Herceptin, Avastin, and the more modern drugs of Keytruda and Tecentriq. However, in the early 90s, we didn't know whether those those antibodies were going to work. We didn't know whether somehow the human immune system would still manage to sniff the little bit of mouse that's left in those antibodies. So we thought we'd better make complete human antibodies just in case. In fact, it turns out the human antibodies we made, I'll come to in a minute, are no better than the humanized antibodies. The immune system does not seem capable of detecting the taint of mouse in the humanized antibodies. But the approach we took was to say, well, if we can't make human antibodies directly, maybe what we should do is to take human antibody genes and create big expression libraries from them. And the way to do that was to take the 2 domains at the end of the antibody, the heavy and light chain domains of all these different antibodies, and to amplify them using the polymerase chain reaction. And therefore, from each cell, so for example, if we look at the cell which is red and white on it, VH and VL regions, this is an individual B lymphocyte. We can amplify the genes from that B lymphocyte, and if we just had a single cell, we could clone the antibody genes for that antibody on the surface of the lymphocyte. But actually, we don't do that. We take a whole population of lymphocytes, human lymphocytes, and we amplify up all the VHs, all the heavy chain domains, and all the light chain domains. We scramble them up, and we generate a huge library of antibody domains. So the question is, we now have these huge repertoires of what amount to antibody fragments, and we thought, well, how do we find out the ones which have got the activities we want, binding to, for example, human cancer, or binding to inflammatory proteins? And we decided to follow the phage display approach, which George has described a few years earlier. And we basically cloned those genes into a phage vector, and we showed that those genes are expressed on the surface of the phage, so as folded antibody fragments. And so on the left, we see a panel in which we can see the you can see the nature of the construction. So you have the antibodies on the outside and you have the genes on the inside. And the right-hand panel just simply goes to show a method by which we identified that the antibody fragments on the phage were functional by taking a known monoclonal antibody that bound to hen egg white lysozyme and testing it on a lysozyme-coated surface. And what we found was, as shown in the lower right panel, is that phage had binding activities, and it was very specific to the hen egg white lysozyme and did not bind to a series of other antigens such as turkey lysozyme or bovine serum albumin, for example. Then we followed the phage selection approach, the affinity selection. George shows, so we could actually show with this model antibody, if we put it in among lots of other phage that don't bind, we can enrich it 1,000-fold on one round of selection. We can take the enriched population, put it through bacteria again, do another round of selection, get a 1,000,000-fold enrichment. So now we put in our libraries instead of this model antibody, and we found that we could, if the library was big enough, in the order of at least 1 billion different antibodies, we could isolate a number of different antibody activities from that library. And we found not only, if we look at the panel on the left, we could find antibodies against foreign antigens. So these are human antibody fragments isolated from cloning the genes out of human B lymphocyte populations. And we could also identify antibodies against human self-antigens. And on the right-hand side, that was in 1994. On the right-hand side, we've got an update from the work of Cambridge Antibody. It was taken over by AstraZeneca. And this is the sort of latest picture. From the same large libraries, one can get antibodies against growth factors, chemokines, ion channels, different types of receptors, GPCRs, cytokines, protease inhibitors, and peptides. So all of these major pharma classes, one can make human antibodies to using this approach. And we can see here that in this slide we show those antibodies that have been made and have been marketed. They've actually gone all the way through clinical trials and they've gone onto the market, and there are at least 60 more antibodies which are in clinical trials at the moment, which hopefully will find their way, or some of those will find their way onto the market. So we can see from the combination of antibody repertoires and from the use of this phage display, we can generate a wide range of different kinds of human pharmaceuticals. So in the last few minutes, I'm going to mention a different approach, which I call bicycles. And this is more recent work, and it relates to work from my lab in the Medical Research Council and from a company called Bicycle Therapeutics, which I set up to take this work forward. If we look at the architecture of an antibody, it consists of loops on a scaffold. But actually, in the course of our work, although the antibody has 6 loops, we discovered that to get good binders, it's not necessary to have 6 loops. In many cases, if you make the libraries big enough, you can get away with 2 loops, varying. All the other loops you can keep the same in the antibody, just vary 2, you can get some very good binding activities if you've got libraries of 1,000 billion, for example. So we thought, well, maybe what we really want to do with this, perhaps if we make something much smaller than an antibody, this might have some advantages. In particular, antibodies have difficulty getting out of the blood. So all the targets, unless they're accessible from the bloodstream, antibodies percolate through very slowly because the antibodies are huge, and it's difficult to get out of the blood vessels. On the other hand, if the molecule is much smaller, less than about 5,000 in molecular weight, it will go through the blood, go out of the vasculature into the tissues. So we thought, let's try and shrink the antibody. And so maybe what we could do is to take 2 loops, just 2 loops, instead of all of this beta sheet and protein here, we could fuse those 2 loops to an organic scaffold. So the core of the molecule is now a chemical, and the loops are bound to that chemical. And this is what I call a bicycle. And the way we do it is we take the phage, we take reactive residues, we take 3 cysteines, which we space along a peptide, and then we react those cysteines with a trivalent chemically reactive species. And this generates on the phage a series of chemically cross-linked peptides. Those peptides are constrained, and in addition, you have 2 loops, and by making very big libraries of them, by making random variations, as George has explained for linear peptides, we can generate even higher affinities than he can get with the linear peptide libraries. So we can get a fit, because they're constrained, you can get higher affinities because you're not having to fight the entropy component. So with these very high affinities, we could get right down to the 50 picomolar range, which is as good as you can do with antibodies. So we can make these targeting modules, and we can make them against multiple targets. So let me explain one of the things we've done. So far we have a targeting agent. Now what we're going to do is to use it to bring in a killing activity. So we link it to a toxin. This shows the bicycle, the targeting entity, a spacer, followed by a cleavable linker, followed by a toxin. So this can be chemically synthesized after selecting the bicycle on the phage. And then we can show that this molecule, if we take a toxin such as DM1, which is a tubulin inhibitor, we can show that this rapidly kills cells that it is directed against. And this shows animal models. In one case on the left, it's a triple negative breast cancer. And it's something which expresses a marker called MT1 or MMP14. And we can show at the high dose of this toxin, which is the orange one, we can see that we can kill, we can essentially destroy the tumor, whereas in lower doses or with no treatment, in fact, the tumor grows very strongly. Again, we can see on the right-hand side, there is another panel there where we can see another model of fibrosarcoma where this marker is highly expressed. And in our animal model, we can show that with the higher doses of antibody, we can completely prevent that tumor from taking off as it would normally do otherwise. In addition, this shows an experiment where we took a well-established tumor. Instead of starting with a small tumor inoculation, we took a tumor which is about 1,000 millimeter cubed in a mouse, and the tumor will normally grow to about 2,000 millimeter cubed, at which point you have to kill the animal because the tumor becomes too large. And so if we look at a dosing schedule, if we look at the picture on the left, the vehicle day 14, we can see the size of the tumor at the point at which we would normally have to kill the mouse. But if we've been giving this bicycle conjugate, we can see the tumor on the next slide, the bicycle BT1718, day 14, this tumor is greatly diminished, and in fact, over a period of time, days 21 and day 28, it disappears completely. So this shows we can blow away even quite a large mass of tumor by targeting this molecule to the tumor, and it releases its payload and destroys the tumor. We've now started to put this into human beings, and so far it's looking promising to the extent that we don't seem to see any adverse effects. It's just in the phase 1 clinical trials. So to summarize, just want to say today I've described 3 classes of drugs: chemical drugs, which are the classical pharmaceutical drugs, biological drugs, particularly antibodies, which is this new, the new wave of drugs. Sorry, I think I'm trying to read Swedish on this thing. I'm trying to stop it. Right, so I shall. Can you stop it, please, for a minute? We just finish this. So we have the biological drugs, which are the antibodies, and which in fact have suddenly taken over. And now what I'm hoping is a third class of drug, which is these molecules which are somewhere between antibodies and chemicals, and in fact have the potential, by combining different features of antibodies, we can evolve them, and we can synthesize them by chemistry, and we can use them as pharmaceutical agents. So I think it's possible that this may be, we believe it may yet be another type of reagent that could be extremely useful for treating human disease. Thank you.
Thank you. Thank you very much. Very elegant. I would now like to present Professor Frances Arnold of the Californian Institute of Technology. Professor Arnold received the Nobel Prize in Chemistry for her development of the direct evolution of enzymes, a field that she pioneered in 1993. Since then, she has made this tool available and versatile, and also applied it into many different beneficial purposes, like developing new enzymes, making use of these enzymes in environmental friendly production of pharmaceuticals, and in the production of renewable fuels, among other things. Please join me in welcoming Professor Frances Arnold onto the stage.
Thank you. Thank you for having us here. It's wonderful to be in Uppsala and see some wonderful snow. Coming from Southern California, that's a rarity for me. I am an engineer who is enthralled by the chemistry of the biological world, the diversity of transformations that biology has invented that lead to life, and the efficiency and selectivity and ease with which nature assembles all these beautiful products of life from simple, renewable, abundant starting materials gives us an inspiration, I hope, for moving forward with chemistry to do the same thing for human beings. Now, of course, the molecules, the marvelous molecular machines that are responsible for all of this chemistry are the enzymes. I wanted to become an engineer of this biological world, and specifically to modify these machines to make things for human beings. Now, how does this enzyme world come about? And everything else, as we know, nature has a remarkable diversity-generating machine that's responsible, a simple algorithm of mutation and natural selection that has generated all of the diversity going back to the last universal common ancestor more than 3 billion years ago. This algorithm that works at all scales, engineers, think of this, one process, one design process that works at all scales of biology, from molecules to ecosystems, is a tremendous inspiration for me as an engineer of biology. Because think of this, I actually started this work in the late 1980s. Many of you students weren't even born then. We knew very little about how the DNA sequence encodes biological function, and certainly not enough to reliably design a new enzyme. So where are we today? let's fast forward through your whole lives, technology has changed tremendously. We can sequence DNA, we can read it, we can write it. You know, you can just type a sequence into your email, send it off to your favorite supplier, get back DNA in the mail. So we can write any DNA we want. We can edit DNA. All the CRISPR-Cas9 amazing technologies that have been developed just even recently, we can go in and modify DNA inside of a living cell. But what we don't know how to do is compose it. We can't even write the sequence of a single enzyme that functions for us. 1980s, we couldn't do it. Even today, it's a challenge. But there is a process that nature used to do this, that nature used to compose the code of life, which to me looks like a Beethoven symphony. And I'm not a very good composer, but I can use this process of evolution. And this is what our challenge was. How do you implement evolution? Of course, human beings have been doing that for thousands of years, making everything from corn to carrier pigeons, lab rats, racehorses, poodles, hairless cats. We have been modifying the biological world for our benefit, not even understanding what DNA is. And we've been making plenty of non-natural things that way. That poodle, for example, would be eaten in Los Angeles. We have a lot of coyotes there. Its fitness is not very high, but it gives us pleasure. And it has functions that we value. Now, of course, in the farmyard or in nature, evolution is limited. Nature can explore a limited diversity of recombination. You smoke a few more cigarettes, you might get a few random mutations. But in general, monkeys go with monkeys, worms go with worms. You're not going to cross the two. You'll have pretty ugly children if you do that. But now that we can make any DNA we want, in the test tube, we can have 3 parents. We can combine DNA from many sources. We can have 33 parents. And this is the challenge, right? It was to figure out what are the strategies that will enable us to breed molecules in the laboratory, in the test tube, on a time scale, for example, that a graduate student could get a thesis done, and without 10 to the 9th graduate students. And we developed an algorithm, which I won't go through in any detail, but basically it's an iterative process of mutating the gene, screening for what you're interested in. Most of those mutations are deleterious, and iterating when you find something good, a small change with a small number of mutations, and iterating on that until you create a catalyst that serves your purposes. And maybe this is an easier way to see it. Nature creates enzymes. They have a biological function. Often they're under natural selection, and they're very good at what they do in the context of the organism that has invented them. But when you ask it to do something for you, it's generally less enthusiastic. So you drop in fitness, but through this iterative optimization process, you can evolve an enzyme for a completely new task. And we were thrilled to find out that this simple random uphill walk optimization works very well on timescales that matter. And today, you might be surprised to know that many of the products that you use in your daily lives are either containing enzymes. Your laundry detergents have lots of enzymes that have been optimized to work in a washing machine through directed evolution. Drugs are manufactured using enzymes, replacing toxic chemical processes that produce a lot of waste, or that use precious metals whose mining leads to tremendous environmental degradation. And many other industries use that. Now, I wanted, in the last several years, to take advantage of the fact that when you have this fast forward of evolution, we can now explore the universe of possible proteins that were never relevant to biology. Now, biochemists, I insult everyone equally here. You're in that little green space of all the proteins that have ever been explored by nature. But the space of possible proteins, essentially many orders of magnitude bigger than the size of the universe, that's my playground. And I can use these evolution processes to go and explore. We can make enzymes nature would never care about. Sometimes humans don't even care about them. But we can do that with this evolution. And I'll just give you one example, because this is a short lecture today. But if you consider something like a carbon-silicon bond, something that humans have used for many products, there's probably 50 products in this room that contain carbon-silicon bonds, from things that you put in your hair to the earphones in your ears, caulk sealants. But nature never made carbon-silicon bonds. You can't find that in the biological world, or at least you haven't yet found it, because there's no known enzyme that assembles that. For whatever reason, nature chose not to develop that. But if you go to the biological world and ask, can you do this? Can a protein catalyze this reaction? We discovered that a cytochrome C from a hot, salty pool in Iceland, we just pulled it from a database, catalyzes the reaction between a silane and a carbene precursor to form a carbon-silicon bond. We had a chemical hypothesis that this could be catalyzed by an iron catalyst, even though a human being had never shown that before, that iron could do this. Now, you may be surprised by this protein because its native function is electron transfer. Oh, and by the way, it's very stable because it comes from this very hot environment. It is so stable that you can boil it. That means even a chemist could use it as a catalyst. And it does this reaction just as well as the best human catalyst based on a precious metal, just this native protein. Of course, nature never cared about doing this reaction, but enzymes are they're like graduate students. Enzymes have multiple capabilities that are not being used at any one time. And proteins, if you're smart, you can look at them like a chemist and say, even though this protein does electron transfer, maybe it could catalyze formation of carbon-silicon bonds. And then maybe it could evolve. Now, if you know something about cytochrome C, you'd say, well, Francis, that looks a little bit off to me, because every chemist knows that this cytochrome C is coordinatively saturated. All the sites on the iron are occupied by ligands in this protein. So where does the chemistry come? And in fact, if you roll a little ball over your model, and this is the actual crystal structure of this wild-type protein, you find that there's no volume for an active site. But you know what? Nature doesn't care what your calculations say, or your view of what a protein can do, because this protein has many other capabilities than this static representation shows us. They're a little bit more complicated than that. And in fact, not only can it do this chemistry, but it evolves. So we could take the wild type, WT, which has a little bit of activity, kind of like human chemistry. It's pretty poor, But it evolves and it improves under this process of evolution, so that after just 3 generations, we could make a catalyst 40 times better. And we could make catalysts that are 400 times better than anything a human being has been able to do. And we can reverse engineer the products of laboratory evolution, because we want to gain knowledge from these experiments that tell us what really does matter when you're making a new enzyme. What can we learn from the mutations that are required to do this? And so we see in this example that with just 3 mutations, 3 changes in the sequence of the protein in the active site, a loop has flipped out and it has carved out a volume for chemistry to take place. No one could predict this. No one could, it's very hard to explain how those mutations did it. We can see the result, and no one can predict that. And furthermore, it has become very dynamic, which allows substrates to come in and out. So we learn from the experiments as well. This works inside of living cells. You can just express this new gene inside of bacteria, and the bacteria will take up the substrates and form these new bonds, giving us a whole new capability that you can add to the chemistry of the biological world. So when you think about it, and this is, I want you to think about where we can go in the future, all of the chemistry of the biological world that we know in making carbon bonds to some other element. Carbon likes to bond to hydrogen, nitrogen, oxygen, sulfur. There's a few bonds to halogens, a few other metal carbon bonds, but it's really a very limited set of the elements in the periodic table. Human chemistry has gone well beyond that. Humans have invented a great deal of chemistry that biology, as far as we know, has never done or has no reason to do, or we haven't yet explored. But in just 5 years, using forward evolution, we've added both boron and silicon to the chemistry of living systems. So now we can go on and modify those new molecules using metabolic engineering, for example. And I think there's a whole world of the periodic table that's left to be explored. We're just at the beginning of this, and I find this in a very, very exciting time for chemistry, because we have this algorithm for optimization, but I've also shown you that it's a process for innovation, for making whole new chemistry in biology just by recognizing the ability of biology and the diversity of biology to do things that we can be surprised by. What does this mean? It means that by marrying what we know about evolution with 100+ years of creative human chemistry, we can now break down those barriers and implement chemistry in the biological world with these clean, green chemical processes in a whole new way. So with that, I bring you some sunshine from Southern California. I thank my current group members and my many former group members who do all this work, and thank you for having me.